We are recording the webinar for a presentation on the LTER YouTube channel, which you can find at US LTER, along with lots of other previous LTER presentations. I'm Marty Downs. I am the director of the Long-Term Ecological Research Network Office, which is the sort of collaboration hub for the LTER network, focused mainly on synthesis, but also on education and engagement across the LTER network. This is an LTER meeting, and as such, we abide by the LTER Code of Conduct. If you feel that the Code of Conduct has been violated in any way, you can reach out to me or to any member of the LTER Executive Board. And those are linked in the Code of Conduct that I've shared in the chat. I am really excited to present today two of our current synthesis working groups. One is a longer-term synthesis working group, the cross-ecosystem synthesis, exploring consequences of consumer loss on community variability is the title of their presentation, coming from the Consumer Absence Generates Ecological Dissimilarity Working Group. Totally win the acronym and the logo competition for that. And we'll have Kelly Speare presenting, Jamie McDevitt-Irwin, who's the co-PI of the working group, was unfortunately unable to attend at the last minute today. So Kelly's going to present for that working group. We'll have some time for questions and answers after Kelly's presentation, and then we'll move on to integrating above and below ground community data to understand temporal dynamics and responses to global change. This is a working group that actually came out of the LTER synthesis skills for early career researchers course. This course applications are open now. So really encourage you. I'll drop a couple of links in the chat after we get going, but this was, I think, a great experience for the people who participated in 2024, 2025. We're offering it again this time just for the fall of 2026. So encourage folks who are able to commit a little bit of time to this to put their applications in. This is also a really nice opportunity to say that the network office is planning on funding new full synthesis groups. So those are synthesis groups that run 3, sometimes 4 meetings. That application is also open now through September of 2026. So we have a nice long window for people to develop project ideas. If you're inspired by some of the presentations today and want to gather a group, you can feel free to reach out to me at the network office. I'll also share some of our synthesis resources. And with that, I will hand it over to Kelly.
All right. Thanks, Marty.
Sure.
All right. Let me just share here. All right. Can everybody see my screen okay? All right. Thanks for the introduction. So we are the CAGED working group investigating how consumer absence generates ecological dissimilarity. And we're really interested in these patterns across both aquatic and terrestrial ecosystems. And so Marty mentioned this at the beginning of the call, but I'm one of the co-PIs on this work, along with Jamie McDevitt-Irwin, Sally Kerner, and Hillary Krumholtz is our information manager, but she's part of our leadership team. And so I just want to say I'm presenting today on behalf of all of us, and unfortunately Jamie couldn't be here at the last minute. So I want to give you just a quick overview of our working group team. So we have three co-PIs on our project, and then we have working group members across, I think it's 7 LTER groups, both aquatic and terrestrial ecosystems. So thus far, we've had 2 in-person meetings. We had a first kind of leadership team meeting at the beginning of 2025, and then we had our first full big working group meeting at the beginning of last summer in May. So this is a picture of one of our excursions at the end of our working day, just chatting science outside in Santa Barbara. Um, so the overarching question about our that we're addressing in our work is we're synthesizing existing data across aquatic and terrestrial ecosystems to evaluate how changes in consumer pressure alter community variability across space. And so when I talk about consumers, I mean both herbivores, so things that eat plants, and predators, things that eat other animals. And so we're really interested in kind of the commonalities that these animals might have and their effects on ecosystems across aquatic ecosystems, which encompass freshwater systems like lakes and streams, and saltwater ecosystems, so intertidal systems, subtidal marine ecosystems, and across all variety of terrestrial systems. So kind of one of the motivating ideas behind our work is that consumers broadly are declining across the world. So about 77% of large carnivores are estimated to be undergoing population declines across the globe. And we also estimate that about 60% of large herbivores are threatened with extinction. And these large herbivores can play really important ecological roles across a variety of both aquatic and terrestrial systems in structuring community dynamics and what ecosystems actually look like. So like what organisms we find as the primary space holders or primary consumer or primary producers. And so we think that when consumers are present versus absent or really reduced in abundance, that they have different effects on communities. So when consumers are present, you can see that we expect to find low community variability. And when I talk about variability, I mean variability spatially across space. And so we've represented this with this little picture diagram here where we have 3 different coral reef communities. And if you look at the organisms that are holding space in these communities, what we're trying to show here is that they're all very similar to one another. You have some corals, actually lots of corals and a little bit of algae, but across space, they look really similar. And what we think is happening is that, or the prevailing hypotheses are that both predation and herbivory act to reduce the species pool. So they allow fewer species to be able to persist under high consumer pressure. And they also limit prey growth rates. So by feeding on corals and algae in this exemplified system here, they keep any one group of organisms from kind of taking over. But we expect to see different patterns when consumers are greatly reduced. And so when consumers are less abundant, we hypothesize that community variability is going to be higher. So in this picture, we're depicting one community that has really abundant macroalgae and another community that has really abundant corals and another community that's kind of mixed. And so the point of this here is that these 3 different communities we're showing are really different from one another. And we call this high community variability or high variability across space. And so the prevailing hypotheses about why this may happen is that when consumers are less abundant, we think that priority effects probably have a much stronger effect. And so by priority effect, I mean things that happen really early in succession. So the order of arrival of species can have really profound downstream effects on the community that develops. And also we hypothesize that when consumers are absent, a larger species pool is able to exist when consumers are not having such strong top-down pressure on these communities. And so these can lead to like really different community development under the same set of environmental conditions. And so these questions were really motivated by work that both co-PI Jamie McDevitt-Irwin and I have done, both in coral reef ecosystems. So we saw very similar patterns when we excluded herbivorous and corallivorous fishes from two different experiments that we had done in our past work. And so what motivated these questions is asking, is this pattern generalizable across habitats, regions, and taxa? And there are examples of this across the literature from other systems, other terrestrial systems as well. But we're really curious if this pattern where reduced consumer pressure can really increase the variability of communities across both aquatic and terrestrial ecosystems. So our primary questions are, how does the loss of consumers influence community variability? And does this effect vary with ecosystem type? And then our second question is, how does the impact of consumers on community variability vary with traits of the ecosystem, such as ecosystem diversity, productivity, latitude, or consumer taxonomy? And so to accomplish this, we have been collecting data from both aquatic and terrestrial ecosystems from existing experiments that exclude consumers from the habitat. And so these types of experiments usually involve building cages, and we talk about them colloquially as cages, but they're really exclosures. So they're fences or boxes or some other kind of apparatus that actually keeps organisms out of part of the habitat. And so by excluding the consumers from the habitat, we can understand what kind of influence they're having on the community. And so we scoured the literature looking for other examples of consumer exclusion experiments. And so for them to be included in our synthesis work, they needed to exclude consumers and compare to some kind of a control, that allowed consumer access. And so this is often, we call it like a cage treatment or a consumer exclusion treatment and a control treatment. They also had to measure community structure and abundance. So they had to, actually quantify the abundance and diversity, or like abundance of different organisms that are in these experimental plots. And we needed them to have a minimum of 4 as a sample size. So they needed to have at least 4 replicates of the consumer exclusion treatment and 4 replicates of the control treatment for them to be included in our study. And another requirement that we decided to include was that these experiments needed to be conducted in the wild, in field habitat. So we didn't include things like mesocosm experiments where they're experimentally enclosing organisms and manipulating communities that way. We only considered studies that were conducted in the field. And so this is a map here showing the spatial distribution and spread of the data sets that we've included in our study. And so in this map here, we've denoted aquatic systems with the blue dots and terrestrial systems with the orange dots here. And so I want to point out that these spanned the globe and across a really broad range in latitude, both for aquatic and terrestrial ecosystems. So when all was said and done, we had about 109 data sources. So data source is like one individual source that provided data to us either through personal communication or through an existing study or data repository. But then of those 109 data sources, we split some of them up because oftentimes a study will conduct an experiment in multiple locations, say spanning a latitudinal gradient. And so we considered those separate experiments as long as they had sufficient replication at each site. So that's how we go from 109 data sources to 300 different experiments that have different metadata associated with them. And so when we break that down to aquatic and terrestrial systems, we had about 88 experiments from aquatic systems and 212 experiments from terrestrial systems. And so I just want to point out that although these questions were motivated by our experiments and past work in marine systems, these experiments are really much more common in terrestrial systems, and that is reflected in our data. But we have done a lot of work to go back through the literature and kind of beef up the representation from aquatic systems as much as we can to try to even it out a little bit. And then within aquatic and terrestrial systems, we have experiments from 13 different ecosystems. So our aquatic systems were dominated by subtidal systems, so things like coral reefs or seagrass meadows, while terrestrial systems were dominated by grasslands. So there's tons of these experiments in grasslands across latitudes and across regions of the world. So super cool. So to evaluate how consumers are affecting community dissimilarity, for each of our experiments, we calculated a metric known as beta dispersion. And so if you're interested, this is a metric you can calculate from the vegan package. It's called the beta disper function. It's actually really easy to use. So beta dispersion is a measure of dissimilarity from individual observation units and how far they are to their group centroid in multivariate space. So like if you plot an ordination, how far is each of those points in, say, the cage treatment from its group spatial median or centroid in space? And so the farther those points are on average to their group spatial median, that's considered more dispersed or more community variability versus points that are really close together represent communities that are all quite similar to one another. And then so to analyze our data, we have run a series of beta regression models, and we've included experiment as a random effect in our models. We've also accounted for gamma richness and sample size because we expect that gamma richness and sample size will both likely increase beta dispersion. And so with gamma richness, you might expect that places that just have more species richness have potential for greater community dissimilarity versus locations that just have a couple species in the species pool. So the next couple slides are showing you some of our results, and just to orient you to these figures, so I have beta dispersion, that metric of community dissimilarity on the y-axis, and caged versus uncaged on the x-axis. And so up on the high end of the y-axis, so high variability in community dispersion are high beta dispersion values versus low beta dispersion values represent low variability in community composition. And then caged and uncaged. So just to remind you, caged often means that there's very few consumers versus uncaged means that there are many consumers or whatever ambient level of consumer pressure there was. And so when we look at our data, just comparing caged and uncaged across all of the studies that we included, we really don't see an effect of consumers on beta dispersion. And this isn't totally surprising. We think that there's probably some other more nuanced things driving these results, but just when we look at patterns overall, we really don't see that consumers have an effect on beta dispersion. But when we break it down to aquatic and terrestrial systems, so now I have these faceted for aquatic systems on the left and terrestrial systems on the right. One of the things that we're really interested in is how beta dispersion and its relationship to consumer pressure might vary across latitudes. And I mentioned gamma diversity previously, and so there's this long-standing thought in the literature that gamma diversity or species richness at a site is higher at lower latitudes. So in the tropics, their ecosystems tend to have more species than in high latitude regions. And so we're really interested in how latitude and gamma richness may impact beta dispersion and its relationship to consumer pressure. And so what we see when we look at all of our data for aquatic and terrestrial systems is that they have opposite relationships between beta dispersion and latitude. So in terrestrial systems, we see kind of this expected pattern where there's higher beta dispersion at lower latitudes, and beta dispersion increases with increasing latitude. And so what this means in practical terms is that there's higher site-to-site variability or community variability at low latitudes in these really diverse habitats, but you see sites become more similar to one another at high latitudes. But we actually see the opposite thing in aquatic systems. So in aquatic systems, we tend to see more homogeneous communities or lower beta dispersion at low latitudes and higher beta dispersion at higher latitudes. And the slope of this line is not as strong as it is in terrestrial systems, but this is a significant relationship. And we also see that consumers significantly influence how beta dispersion is related to latitude. And just to kind of circle some points on this figure, so we see at lower latitudes, consumers actually homogenize communities, whereas at lower latitudes, both for aquatic and terrestrial systems, consumers homogenize communities, so they make them more similar. But at higher latitudes, effect is opposite. So consumers increase beta dispersion at higher latitudes, which is really kind of counterintuitive and a little bit confusing. And so we're still thinking about like hypotheses for why we might see this pattern. But overall, we don't see like a really strong effect of consumers either at higher or lower latitudes. But because our sample size is so big, this does shake out as being a significant effect. So our future steps for this work are work that I just talked about today is really going to be a first big synthesis paper that we're pushing forward in the next few months. But our future steps are to explore how spatial scale and plot size may influence these results, and also to explore how these patterns may vary with ecosystem types. So coral reefs versus grasslands, how these patterns may differ across different ecosystems, and how they also may vary with consumer taxonomy. So you might expect that different types of consumers might have really different effects on communities. And so this is going to be a second paper where we're really digging into these patterns more and the drivers of the patterns that we're seeing across these broad spatial scales and across latitudes. Um, and so with that, I want to really thank the LTER network for funding this work. This has been such a rich and wonderful collaboration that we are so thankful to be part of, and we've really, really enjoyed. I can say this on behalf of the whole leadership team that this has just been really productive and a super fun way to build this collaboration and to explore these patterns with other LTER collaborators. So with that, thank you again, and I'll take any questions you have.
Thanks so much, Kelly. Um, we, you can, uh, sorry, participants can just unmute and ask questions or raise your hand. That's fine. Um, I guess I'd be curious. You have quite a lot of data sets that were incorporated. What are the most common sort of other sources of data that you ran into? Are they just individual studies or are they related to other
Other
networks?
sources yeah, yeah. So kind of the first pass that we took was digging through EDI and searching the LTR data sets. We knew going in that there were certain other LTRs that had done a number of consumer exclusion studies. So we looked for those first and looked through EDI a lot. And then we also just started combing through the literature and we found a ton of data sets just from existing studies, published in places that required, data sets to be published online. So we used those. And then we also went through and found studies that didn't necessarily publish their data along with the paper. And we reached out to a whole bunch of authors of papers and asked, hey, we're doing this synthesis working group. Would you be willing to share their data, share your data with us? And we got a ton of responses from that. And so, yeah, it's been really fun to communicate with people who have done studies like this in other systems and incorporate their data. So I would say a wide variety of different data sources.
Neat. Thanks so much.
Yeah.
Ashley.
Yeah, thanks, Kelly. This is super cool. I have a couple questions. Actually, my first was going to be essentially what Marty asked, is I was really interested in hearing more about your data discovery process. And I guess could you talk a little bit more about when it came to just like looking through published papers? What did you use for that? Did you use Google Scholar? Did you use, like what was your process for that?
Yeah, I mean, once we found kind of the obvious low-hanging fruit on EDI, we used Google Scholar a lot and
Hmm.
had some key terms that we were searching. So we searched like herbivore exclusion, consumer exclusion, a variety of key terms to find things that, popped up easily when we searched those. But we also had quite a few studies that didn't necessarily come from those big searches and were kind of more targeted searching for individual ecosystems. So if I don't know if I might be able to go back here. This might take me a while. When we were looking at kind of the distribution, both across aquatic and terrestrial systems, I mentioned there's just so many more terrestrial systems than aquatic systems. And, there have been synthesis projects on terrestrial systems. There's a working group that Sally led that, brought a whole bunch of terrestrial systems that they had already integrated into our workflow. So that was great. So a lot of our searching was really focused on finding additional aquatic systems because they were so much less represented than the terrestrial systems. And so we started kind of tallying up how many studies from different types of ecosystems we had early on. And we were just trying to get any kind of replication we could achieve within an ecosystem, because we knew that down the road we were going to want to think about effects, within an ecosystem or how they might vary across ecosystems. So we were searching for like, okay, can we find any more in coral reefs, or can we find any more in salt marshes? And
Right.
we did quite a bit of like emailing colleagues, like, oh, I have this friend who does community ecology in salt marshes. I wonder if he knows of anybody else. And so we did a lot of that kind of targeted tracking down to try to find anything else that might not be on our radar and might not like have those keywords that we were really looking for.
Yeah, no, great, thanks. We had some good luck with like Google data sets as well, but then we just decided, because with the Spark timeline, we just pulled back a ton, but those are things that we're thinking about. I
Yeah,
actually
I
have
will
another.
say that doing this has taken a really long time. I mean,
Yeah,
we
yeah,
did kind of a first pass of data discovery, and Nick Lyon, who is just an absolute wizard and a hero of our data
yep, yep.
pipeline, helped us a ton. But I mean, even over the last more than a year, we are still continuing to integrate data sets. So we did kind of a first pass of analysis with what we had, and then keep doing, additional runs of it as we integrate more. So it
Yeah.
definitely takes a long time.
Yeah, that makes sense. I have another question, but maybe I'll stop for now and let other people pop in if there are other questions.
Fine, fine with me. It doesn't matter.
All right. Okay, I'll go ahead. I was just curious about how you thought, and I'm sorry if I missed this, at the role of seasonality, maybe with some of the results that you saw, like with the latitudinal patterns
Yeah.
and it's kind of how it's mediated in aquatic versus terrestrial systems.
Yeah, so this is something we have increasingly been thinking about, especially for our aquatic systems, because, for aquatic systems, especially in high latitude, they're almost never sampled in the winter. And so we're
Exactly. Yeah.
wondering if some of the patterns we're seeing could be really different depending on, like, okay, well, most of our high latitude aquatic systems were sampled in the summer because they have sea ice in the winter. So that is something we're thinking about. And also, I mean, the data are what they are. We don't have the ability to get data in times that weren't sampled, but at least to think about like why we might be seeing that pattern, and whether there's reasons to think the pattern could be different if there were data available during the opposite season. And
Yeah.
there was a study it's a microbiology study in like marine ocean microbes, that like the seasonality pattern actually can reverse that latitudinal gradient in biodiversity and gamma richness. So definitely on our radar.
Yeah. Yeah.
Yeah, not sure what we're gonna do about it, but
No,
it's
I know it's
good
tough.
food for
And
thought.
yeah, and the reason I ask is so I'm a microbial ecologist, and like I work mostly in salt marsh systems, and I'm like I want to start sampling in the winter, like under the
Yeah,
snow and the ice, and it can be completely different. But yeah, this is really cool. Thanks so much.
thank you. Thanks for your questions.
Really fun. Thank you, Kelly. Um, I imagine Kelly would be willing to take additional questions from folks in the chat if they occur to you, but we're going to move on to the next working group. This is the above below ground working group. Oh, Uh, and I think Ashley's going to kick us off.
I was muted. How does that look? Can you see my screen?
Looks
Okay.
great.
All right, awesome. Yeah. Yeah, so my name is Ashley Bulseco, and I'm here with my colleagues Kaitlyn McKnight and McKinley Nevins, and we're here as representatives from our Spark working group that's focused on integrating above and below ground community data with the goal of really trying to understand ecosystem dynamics over time in response to global change. And so before we get started, I wanted to take a moment to introduce the team. So this proposal, as Marty mentioned, actually emerged from a group that formed as part of the Synthesis Skills for Early Career Researchers training program offered by the LTER last year. And so they're currently recruiting for the next cohort. So I had to toss it up in here. So the QR code will lead you to the application. Deadline is March 31st. And as part of this program, Abigail, Francis, Mariti, McKinley, and I learned how to conduct synthesis science, maybe even more importantly than that, team science. And so kind of the ideas that we'll present today came from that experience and from discussions we had as part of that Seeker program. And so together we submitted a Spark proposal, and we brought together some new folks to expand our group who bring in expertise across disciplines and across different career stages that would allow us to answer some of the pressing questions that we think in ecology related to above ground plant and below ground microbial communities. And so that's the focus of our team, is to really understand associations between these plant and microbial communities. Why these plant and microbial communities are tightly linked. They form the foundation of ecosystem processes. So their interactions, I don't know if you can see my pointer here, but their interactions regulate the fluxes of energy, of nutrients, and of matter through ecosystems. They influence processes like carbon cycling and nutrient transformations and primary production. And so because of this, these associations influence ecosystem functioning. However, these kind of interactions are dynamic over time. And so if we study plants and microbes in isolation, which very often happens, just many times because of logistical reasons, it's really difficult to predict how ecosystems are going to respond to environmental change. So we need to understand these kind of plant and microbial communities in tandem to try and understand ecosystem dynamics and kind of their influence on each other. So kind of our team, again, this is a Spark group, so we just started last fall, and we're meeting for over the next year. And so we kind of came up with 3 parallel projects to try to get at these knowledge gaps. And so the first is ongoing. So data discovery is we're trying to explore kind of the characteristics that existing data sets have, and to think about ways that we might be able to expand on data that are being collected. And so I'm going to kind of touch upon that in the next slide. We also have a case study that's going to take some of this empirical data that emerges from the data discovery process to really dig into questions that try to quantify plant microbe synchrony across ecosystems. And so Kate's going to take that on in a couple of minutes. And then finally, we wanted to kind of take a step back. We felt there was a need to really think kind of more conceptually about a framework, more holistically about plant microbe interactions. And so we're thinking about this in a perspective piece that McKinley will end on today. And I want to kind of note before we're moving forward that all of these projects are very much ongoing, and we'd be happy to take feedback or ideas from you all on any of this. And so at the end of today's talk, we have kind of QR codes that will offer our contact information. We really encourage you to get in touch if you have ideas and if you have data sets that might help with some of these questions. And so I just wanted to quickly talk about the data discovery process, which again is an ongoing effort. We really wanted to try and identify data sets that satisfy our criteria for this case study. So again, this was a Spark working group, so we have about a year to try and accomplish some of our questions. And so our initial goal was to really, again, just try and identify data sets with these criteria. And so we started with the Environmental Data Initiative, so EDI, and we used these 2 kind of queries, microbe and biomass and microbe and community, which gave us a total of 449 data sets to go through. And we basically retained data sets based on of these criteria. So the first kind of filter was, do the data sets have at least 6 years of data? So we needed a long-term data set to be able to answer questions that got at like temporal dynamics. And Kate's going to talk a little bit more about that in a second. And then we also were looking for data sets that had both above ground plant data and below ground microbial data, either biomass data or community data. And so we flagged those that we weren't really sure about, and that allowed us to further explore those data sets, and it left us with a set of data sets that we felt pretty confident that we could move forward with the case study. And so we learned a few things out of this process, again, a preliminary kind of data discovery effort. And the first was, if we looked at the number of studies that came out of just these queries alone, that the vast majority of them, probably unsurprisingly, but the vast majority of them were short, so like 1 year or less data sets. But that also, there was a good handful of those that were, upwards of 10 years or more. And so that was an interesting distribution to see. And then the second thing we learned, which again was not really surprising because we had been kind of more informally doing data discovery before as part of Seeker, is that there actually weren't many data sets that were going to satisfy our criteria. So these maps are showing the results of these 2 queries just within the LTER network. So these are those that came back from microbe and biomass on the left and microbe and community on the right. And then if I remove the ones that didn't meet our criteria, these were data sets that we flagged that we thought would have above and below ground data. And then these were the data that we were left with that had above and below ground data for at least 6 years. And so there's 5 data sets we're confident in, and all of them are biomass and none of them are community. And so we have great data sets that we're really excited to work with, and we can address some interesting questions with that Kate's going to get into in a second. But what this also made clear is there's a very clear opportunity for us to expand our efforts to try and collect data for both plant and microbial communities at the same place and at the same time. Okay, so I'm going to hand it over to Kate, who's going to continue to talk about the case study.
Awesome. Yeah, so one way we can study associations between plants and microbes is by investigating how synchronously they fluctuate together over time, which we commonly define as ecological synchrony. And so ecological synchrony, especially community synchrony, has gained a lot of traction because of its relationship to ecosystem stability. So from the plant world, which I'm showing here in this green example, we have this general understanding that when species are in a community are synchronous, this can lead to instability in, say, above ground biomass or productivity over time. But we don't, however, have a strong grasp on how these patterns might play out when we're considering synchrony between two kingdoms, say, plants and microbes. But this can be especially important when we know that the connectivity between these two groups is critical for whole ecosystem functioning. And so, So to quantify patterns of synchrony between plants and microbes, we required a time series have at least 6 years of paired data. And that was for 2 reasons. So most synchrony metrics are based on covariance or correlation between 2 time series. And if a time series is too short, the covariance estimates can become quite noisy, or correlation can have very high variance, or maybe just 1 year is dominating our estimates, and this can make them really unstable or unreliable. But also we were constrained again by those number of studies that had more than just 1 year of data. So requiring a longer time series would have made us exclude too many potential data sets. And we didn't want to estimate these general patterns from just a few sites. So that's why we required at least 6 years of data. And then, so luckily we do still have at least 5 sites from our data team's exhaustive search. And these 5 sites have that 6 years of data of appropriately paired data between plants and microbes. And so these will allow us to begin addressing some foundational questions as a case study. And so with the case study, we hope to evaluate just how common patterns of synchrony between plants and microbes are, how these patterns might vary across environmental gradients, but also if this variation depends on vegetation or ecosystem type, since those 5 sites kind of encompass multiple ecosystems and vegetation. And so it's also very important to note that when we're using data from the LTER network, we're investigating these questions with biomass as our community metric. But if we incorporate additional paired data from NEON, we can also explore these questions through changes in community composition over time. So this will allow us to gain a thorough understanding of synchrony between plants and microbes in these 2 different ways. But while this case study serves as a way to answer these foundational questions on synchrony between plants and microbes with empirical data, there are still a ton of other applications and considerations for when we're exploring these ideas, like how they relate to ecosystem stability, like I mentioned before, which McKinley will now dive into with the perspective piece.
Awesome. Thanks, Kate. Yeah, so we've also been working on this perspective. Like Ashley said, we wanted to zoom out kind of to a bigger picture. We've been really thinking a lot about what is the kind of theoretical and mechanistic basis for these interactions. And so we're doing this for ourselves too, to set a good foundation, but we also think it'll be really beneficial for the field. So we've been working with a few additional external co-authors that we wanted to mention here who have been really, thinking about these sorts of dynamics for a long time. And so as Kate mentioned, we've been applying synchrony approaches to plant populations, and more recently to plant communities for quite a while now. But to our knowledge, these sort of mathematical approaches haven't been applied to considering the joint dynamics of like plant microbe communities. And part of that is because there's kind of a lot of tricky considerations here, which I'll talk a little bit more about in a minute. But our goal, part of what we want to offer here is really kind of generating these testable hypotheses for how plant microbe synchrony or asynchrony responds to global change specifically. And so we're really lucky that we have a few great modelers and theoreticians on our team who are doing some theoretical simulations where, along with our kind of empirical case study, we can also test in silico how synchrony can behave under different conditions. So we are manipulating different kind of environmental forcing scenarios, and we can also manipulate different components of the kind of model plant microbe communities. So we can manipulate their species richness and see how their synchrony responds. We can manipulate the kind of degree of coupled dynamics between the two components and see how synchrony responds, which is an exciting thing. And so through that, we will also be able to have these hypotheses that we can test a little bit in this perspective as well. And so, like I said, our goal here is to really offer kind of more of a conceptual basis for how we can think about plant microbe synchrony, and more broadly, just how we can think about and quantify full above below ground responses to environmental change. And so, as I mentioned, there's a lot of different dynamics and considerations to think about here, and that's part of what has made it so tricky for us to think about how to quantify synchrony effectively for these communities, because of course they are operating on very different spatial and temporal scales. And so this is a conceptual figure we've been developing for the perspective paper. And our goal here is to just represent, kind of two different ecosystems responding to two different stressors or kind of global change factors, and then demonstrating how component plant communities as well as component microbial communities could be responding on very different timelines. And we also have here in green bars changes to biomass, and then in orange changes to species richness. And so this is something we're going to be continuing to calibrate as we gather more literature on this. But obviously there's a lot of dynamics that are responding in very different ways. And so this really underlies why we feel that we have to have long-term data to be able to study this effectively. And additionally, why these components need to be sampled in a very tight spatiotemporal kind of matching for us to be able to assess this effectively. Okay. And we've also been additionally doing some other literature review. Actually, Alex Seegers is here. He's the one that has put together this figure. And so we've been thinking, once again, along with having our case study that's actually testing kind of synchrony across ecosystems, we've also been looking in the literature at examples of kind of synchrony and stability relationships. And so we have here 4 different ecosystem examples and looking at how the amount of synchrony between the plant and microbial communities varies, as well as how that might affect ecosystem stability. And if folks have additional questions, Alex, I might put you on the spot later and you can talk about these a little bit more. But we wanted to just provide, at least from what's in the literature so far, some examples of these dynamics. Okay. And then to move it a little bit away from just conceptual into kind of more practical applications, we're going to kind of finish up the perspective piece by highlighting 3 kind of practical areas where researchers or practitioners could benefit from considering our conceptual framework. And so we've decided to focus on conservation, restoration, and agroecosystems because there's very specific goals in all of these practical applications. And we are intending to argue that when you are considering, or when you take the time to consider the connectivity between plant and microbial communities, that you can actually meet your goals better when you're better understanding how those components are interacting and how they are kind of jointly responding to disturbance or environmental variation. Okay. And so, through our data search, we have found, of course, lots of gaps in this data. We're sure there's more out there. And so, like Ashley said, if you are holding on to some data, we would love to talk to you about it. But we want to really find ways to overcome some of these barriers that we've been really thinking a lot about. And so just a few that we're going to highlight very briefly that are kind of the tip of the iceberg of these bigger efforts that we're going to be working toward. The first is that we lack kind of standard methodologies for sampling, right? We see as Ashley said, logistically, it's usually easier to sample plant communities, sample the above ground community. And so we have a lot of plant data, but the microbial data below ground is really kind of the limiting factor. And as I mentioned, because these are responding on really different spatiotemporal scales, we have to have very close alignment of sampling time and sampling location to be able to look at synchrony of these components. So we are actively working on making a set of recommended methods that someone hopefully could pick up when they're writing a grant proposal or establishing a new experiment and be able to have a plan for spatiotemporally paired sampling of these plant and microbial communities. So that's the first step. We also know that part of why this feels sort of daunting to sample both plant and microbial communities is because we all have very domain-specific knowledge and expertise, and most of the time it's only in one half, right? And so our goal is that we can build, excuse me, this more of a network where we can connect the plant folks and the microbe folks, where they could be collaborating in initiating projects or in adding a sampling component to an existing project, where then we can have both the plant and microbe being studied together. And then finally, our search so far has been primarily focused on EDI, as Ashley said, just based on kind of timeline constraints. But we know in general that there's a lot of this data that exists, but it's very diffuse. And right now, there's not really a lot of crosstalk between different groups or different networks that are collecting this sort of data. And so along with these recommended methods, we also are really trying to think about the ways in which this data is already being collected by existing networks, because we would love to be able to kind of have these fit together better in a way where data that's already been collected can be used by more people. And then data that will be collected in the future can be kind of plugged right into those existing data structures. So this is very much an area of a lot of work right now and into the future. And we would love feedback and ideas, especially on how to make this very functional. So on that note, I will pass it back to Ashley.
Yeah, so I'm just gonna wrap it up and say, yeah, as McKinley said, this is very much a work in progress or works in progress. We're really interested in getting feedback or talking to you about data that you may have. Of course, we know there's more data out there, but it's just a matter of getting it and trying to figure out, making time to get it. And so I'm going to come back to the slide in a second, but just also wanted to throw up our acknowledgement slide. There's lots of folks to thank, especially the LTER and our experiences as part of Seeker as well. And so I'm going to put it back on this slide. These are QR codes to our website on the left, and then on the right is kind of like more of a form. If you just want to express general interest in kind of chatting about some of these ideas, we really encourage you to get in touch. And that's all we have. Thank you.
Thank you, all of you. I have a quick question, and then folks can chime in with additional questions, I'm sure. Um, do you did you have a criteria for how close together sampling needed to happen of the microbial and the plant communities? Like, is there did they have to be at exactly the same time? Did they have to be kind of part of the same sampling trip? Did you have a feel for that?
I'm just going to start by saying for the EDI search, we didn't, mainly because there just weren't a ton of data sets to begin with. But I will say that in our in-person meeting, we spent a good amount of that time talking about the neon sampling protocol, and thinking about trying to figure out which of those samples we wanted to use, which had a lot to do with temporal matching and spatial matching. I don't know, McKinley, is there anything we should add to that?
Um, yeah, definitely from neon. Just in the way that they are organized, their soil cores for the microbial communities are sampled in kind of a rotating way within the same I think it's the 10 by 10 meter plots of the neon like plant community sampling. So of course, all of us soil folks know that that variation is at a much smaller scale than 10 meters. But in those subplots, we could align things relatively quite closely. And then temporally, yeah, neon is sampled during the growing season. Sites with a longer growing season are maybe visited twice. That doesn't always necessarily guarantee that the microbial community was sampled twice. But from those, we're still able to match up a ton of plant plots and a ton of microbial cores with a lot of replication. And Jeff Zahn, our kind of bioinformatics guru, has been running all of the microbial sequence data for bacteria and fungi. And we have like over 1 million new ASVs that have been assigned that are taking over the high performance cluster at William and Mary. So we're going to have that data in hand very soon, and we'll be able to trim down to the closest like spatiotemporal pair that we can get for that. Yeah.
Other folks have questions? I'll take the opportunity to just take a minute to explain the relationship between Ashley referred to Spark working groups, which are only funded for one in-person meeting, and the full working groups, which are funded for 3. And these guys have done amazing work, partly because they started in the Seeker course, but the idea behind the Spark working groups is to sort of use them to pull an idea together to sort of test whether you can find the kinds of data that you need, the kinds of collaborators that you need, whether the idea that you have for a project really has legs or not. And so I would encourage folks to submit. This current round of working groups is 4 full working groups. We'll have another round next year that is for Spark working groups, and sort of be bold with your Spark ideas. You don't have to know that they are that they're going to work out. You can explore new directions that might be a little bit riskier than otherwise. Any other questions or ideas for data for these guys?
I had a question or just like kind of a comment and a question for the group.
Yeah.
Let me see if my camera gives you more than the white screen. It will not. Okay, we're just going back. Never mind. Apologies for that. So first of all, very cool. This is a subject that's near and dear to my heart. It was nice to see that dot for the paired samples in Southwest Michigan. I'm based at KBS. But I was wondering, to the point of how there's a lot of microbial data in particular that's outstanding. I think that there's been a lot of most of the core LTR executive committees have had someone with a plant focus on them, through like a majority of that tenure. Those are kind of core data sets to the LTRs. And for instance, at KBS, like we have, Sarah Evans is on the executive committee here. She's a relatively recent addition to the executive committee. She worked, on the LTR before that as well. But like our longest term data set, microbial data set is actually coming from Greg Bonito's lab. He's an MSU faculty. He's not associated with the LTR, other than as a co-investigator through other grants that he's gotten. And one of his grad students is working that up through a fellowship, which is pretty cool. But it's also, if that fellowship wasn't there, then even though she's using that data for her own research, she wouldn't have the time to then work it up to make it into a data set that we could have as part of our data catalog, which is where it's going to end up, which is really cool. So I guess if it was wondering if you all have thoughts on like what's the best, like where could we get the funding, or where's the need that the proposal could really then, exhibit that if you put resources towards trying to collate this data across the LTR, you could really get something out of it. Because I do think there's a lot more microbial data across the LTR than what is currently associated with each site's data catalog.
I will say when I forget who had the slide up about each section of the project and each of the obstacles, but a network of researchers across microbial and plant communities sure looked like an RCN to me.
That would be really cool. I think also for the community building and getting early career folks into a network like that. That would be really cool for microbes and
Yeah,
plants. They're not just carbon pumps.
right. Yeah, no, I think we're hesitating because I think it's something we still haven't figured out yet. Like we know it's important, but the actual how we're going to make it happen is challenging. But Marty's point is a good one, and that is like, yeah, thinking about it, at least start with building a community that talks about some of these issues, and then maybe going from there. And so that's something that, McKinley's mentioned is something we're thinking about how we're going to make that happen, and which, initially is going to be something more grassroots and then trying to get funding. But I think also you make another good point that, yeah, we started with EDI because we have this year and we have so many big ideas, but we did need to try and cut it back. And so we've discussed a lot of different iterative approaches that we'd use with data discovery that could hopefully capture some of those things. Just like, what Kelly mentioned, part of that is like just reaching out to people we know. And there's a lot of, especially international collaborators that have really graciously offered data, but we just couldn't really take it into account at this time. But that's going to be really important, is like leaning into our networks, and we were looking into we're thinking about even looking at like NCBI and things like that, but we're holding off on that. But there are certainly other ways that we hope to tap into microbial data, which when it's published, it's required to be deposited, right? So that's something we can
Mhm.
leverage at some point. We just haven't gotten there yet, but that's a really good point. Thank you.
And I'll just add to like, yeah, the like you said, the working up for the microbial data and everything can feel more daunting. It's more specialized. There's bioinformatics pipelines, and along with standardizing the methods for actually going out and sampling, the pipeline you use makes a huge difference for data and whether it can really talk to each other. And so we're also thinking about tools or pipelines that we can be generating that can lower the barrier to entry potentially for processing some of this data, and can also make all the steps along the way be more compatible. And so as a student who figured out my own bioinformatics pipelines, with some help, but a lot of it just grinding, I think if we can offer some tools and stuff that can make it easier for folks to work it up, then that can be a big benefit too, and can help that data get into the public domain faster. So,
No, totally. Those sound like awesome resources, and just yeah, thank you all for for this. Very excited to read the perspectives paper and just really exciting to know that this work is going on.
Yeah, thank you.
Absolutely. Thank everybody one last time and wrap things up.
Cool.
Thank
All
you.
right, thanks everybody.
Thank you both.
Thank you.